Hey guys, so we're going to do session 13 material. This is technically session 14. We're going to do session 13 material, which is all about the myofilaments and the contraction and everything. Sorry, my neighbor sucks. He sucks. Anyway, okay, so this is all about myofilaments and contraction and everything, and we're still going to have a session on Tuesday, and then test prep on Wednesday, and then y'all's test I think is on Thursday. So I uploaded a study with me video, which I think is kind of fun. It helps me motivate if I just put someone up on my screen or like split screen or something, and so then I can study with them, and it's not like me doing it all by myself. So this is just going to be just like a regular session, but I'm just going to teach y'all instead of y'all being here, and then I'm just going to upload it. I know I'm being weird, I'm trying to get comfortable. Okay, so I'm going to share my screen with no one, which is okay because that's fine. All right, so first off, we're going to start here. Okay, so actin, let's see. I can't go up anymore than that. So actin, right, we said is the thin filament, okay? It's across the I band and partway through the A band and anchored to the Z disc. So it's very confusing. Let me draw it for you. We'll talk about that. Right, so then they have a myosin head or myosin tail and a myosin head. So the myosin tail are the heavy chains, right? They're intertwined. So these are the myosin tails, right? The globular head is the myosin tail, and then the myosin head. Natalie, edit this piece out. Edit this out. Okay, so the myosin tail, okay, the myosin tail is these, right? These kind of little squiggly bits, and then the head, right, is that globular head, right? So it's this piece, okay? So don't get really discouraged about this. I honestly don't think you're gonna need to know this. You might need to know this. You might need to know that it's too heavy and for light, okay? But I don't think you need to know that, like, what is what. But also notice that this does add up to 6. So that helps you remember that this is the thick filament, which is also myosin, okay? So then during, those go with us. Okay, so then during contraction, the heads link and form that cross bridge, right? So this head, this head will come up and link up on here, right, and form a cross bridge, right? If this is actin and this is the myosin globular head, it'll come up here and form a cross bridge so that it can pull, okay? I'll show you that later, so don't worry about it, but I need to get rid of all this drawing. Okay, so then, okay, so then the thin filament, right, is actin, right? It's a fibrous protein. It has a G-shaped kidney polypeptide. They kind of like to ask about this. They have a globular subunit. I'll show you that in a second. And then during contraction, let's just watch this video really quick. I think this might just make it easier. The same room. Okay, so this video, right, is stellar. This is the video. Watch it over and over and over again. Okay, so this is actin. All right, so here's our sarcomere, right? So this is little actin. These big globular head pieces in the middle is myosin, okay? And so this is on a subunit, right? So down to the subunits, okay? Because this is myosin, and these are those tails, and these are the globular heads, okay? And this is actin. So this is that pulling up and attaching to the actin piece, okay? And it pulls, and it makes that cross bridge, okay? And then it does this power stroke is what it's called when it contracts and it pulls, all right? So then there are fibers, okay? Okay, so there are fibers on actin. So this is what actin actually looks like, okay? and the fibers are in the way of the binding site for this myosin head. I know we're getting a little bit ahead of ourselves in the worksheet, but I think that watching this video will help me explain the worksheet better to you. So, right, so myosin head wants to come up and be on these actin binding sites, but these filaments and this little blue protein are in the way of that. So calcium allows us to move those out of the way. So calcium is flooded into the muscle cell, okay, and it attaches to troponin, right? So we have two proteins. We have troponin, which is the blue piece, and then tropomyosin, okay, which is the filament that's the filament that is in the way of myosin. So if that's how you want to remember it, that tropomyosin is in the way of myosin, okay? So then calcium comes and it's really slow apparently. Okay, calcium binds to the troponin, right, which allows us to do this. Okay, the calcium binds to troponin, and then that allows us to pull those fibers away, right? It pulls away the fibers from where they are so that it exposes this myosin binding site. Okay, and then we're allowed to have this myosin come up and touch the binding site so that it can make that bridge, cross bridge, and then do the power stroke. Okay, so if you need to watch that piece 100 times, honestly, watch this video so many times. Just watch it a lot because it tells you everything you need to know for your exam. And I think it's pretty user friendly, and the lady's kind of annoying, but other than that, it's an amazing video. Okay, so we're talking about during contraction, right? We twist that tropomyosin together, right? And so then the troponin, right? So then okay, so during contraction, okay, the active site for myosin comes up and we bind, okay? So let's just do, what's this thing? Okay, we're gonna talk about this. Okay, so the tropomyosin, right, is blocking that site. The troponin is what binds to calcium. The troponin binds to calcium and allows us to pull away that tropomyosin so that that myosin head can come up and touch on actin. Okay, so this is a little crude little drawing that I've done. Okay, so this is tropomyosin, and this is that troponin, and tropomyosin is the filament, and the calcium comes. I want it to be. What happened? Okay. The calcium comes and binds to troponin, which allows us to pull this filament away and pull it tighter, right, almost, so that that globular head can come up and bind right here, right? So this is myosin. Sorry, I'm like doing it on a trackpad, so it's like rough. Myosin. Could have typed, I guess. Okay, so this is myosin down here. So I hope that kind of makes sense. Okay, so go watch the video a bunch, and then you'll know what I'm saying. I'm sorry if I'm not doing a good job. Okay, so then we have three modified organelles, a myofibril, the sarcoplasmic reticulum, and we have T-tubules. Okay, so the sarcoplasmic reticulum is a network of smooth endoplasmic reticular tubes. Okay, so just bear with me. I'm going to show it to you in a second. So it's a network of smooth endoplasmic reticular tubes, right, that surround each myofibril. Okay, so it's on the outside. They run longitudinally. Okay, so like this. Longitudinally. How to think about it. Run longitudinally, and SR always just means sarcoplasmic reticulum. I just don't want to write it a bunch of times. Functions in the regulation of calcium, right? And calcium is really, really important because calcium is what binds to troponin, right? That allows us to move that tropomyosin filament, okay? Right, so it stores and releases calcium, okay? So let me show you the sarcoplasmic reticulum. Okay, so how does it do it bigger and then small? Okay, so right? This is your neuron, which we'll talk about in a minute, okay? And then these, underlying this red is this muscle cell, okay? So, we're gonna talk about that. Okay, no, no, no. I'm having trouble with this. Okay, so overlying, right, longitudinally, okay, I guess longitudinal is this way. No, this is latitude. Maybe I wrote it wrong. It runs this way, okay? The sarcoplasmic reticulum runs along the fiber, and it literally looks like kind of like goo, or like it's a network, and it just stores, those little yellow things are calcium. It just stores calcium, and so then the sarcoplasmic reticulum will release the calcium. Oh, it's going to go through the whole, okay. So, okay, okay. Yay, this is happening. We're going to know what happens in a second. Okay, T-tubule, then to sarcoplasmic reticulum, which then releases this calcium. Okay, which is why it's so important. Okay, so then we have the T-tubules, also called the transverse tubules, right? These allow an increase for surface area. These run up and down, which I think is longitudinally. So I'm going to fix longitudinally, right? This allows for the electrical nerve transmission to reach the fiber. So back here, when we have this whole bit, okay, this is the neuron. Let's do this. Okay, so we have this neuron, right? And then the neuromuscular junction, and then these are the muscle fibers. But if we look closer, I know I'm doing a lot of like back and forth, right? We have the sarcoplasmic reticulum in here. These, right? The red piece, you can see the myosin and actin in there, and then you have this blue piece, which is the T-tubule. Okay, so the T-tubule allows the it allows the signal to come down the neuron, and then it goes through the T-tubule as an electrical impulse, okay? And then it goes into the sarcoplasmic reticulum. So the T-tubule allows that electrical impulse to go all the way down the nerve fiber. So here. So instead of oh, right, this is your neuron. Ooh, this is your neuron, okay? Ooh, this is your T-tubule, and then the blue is the sarcoplasmic reticulum. So as the nerve impulse travels down this, right, it has to get to all of this, everything over here and over here. So the best way to do that is to go down this, and it can transmit it everywhere, okay? Instead of just going here to here to here to here, okay? I hope that was not like horrible, but we'll see. Okay, so this allows the electrical impulses to be transmitted. That's like the big piece. And then it increases your surface area, right? So instead of just like jumping all around, it goes down the T-tubule so that it can just spread, okay? So then we're going to talk about our muscle anatomy. So why is this? Okay. So our largest piece over here, right, that's surrounded by the epimysium, okay, is your whole muscle, okay? The whole muscle. I'll do a picture in a second. And then we have the fascicle, which is surrounded by the paramysium. Then we have the muscle fiber, which is surrounded by the endomysium. Remember, epi means outside, peri means between, and endo means within. Okay? Okay, I know this endo is supposed to go here, so don't get confused, okay? And then we have the myofibril, right? Myofibers are within muscle fibers. And then we have sarcomeres, which is like so important that these are the functional unit. They're the smallest contractile unit. Don't let them trick you and say muscle fibers or myofibrils. Those are not the functional unit. They are not the smallest unit to contract, right? It's a sarcomere. And then we have our myofilaments, which are actin and myosin, okay? So the sliding filament model is talking about how the shortening happens and the active cross-bridge generation. So it's just what we've been watching in the video. It's just talking about how the sarcomere shortens. Also, let's go look at the let's do this. So, right, so we have the whole muscle, right, epimysium, we have the fascicle, peri, and then we have the myofilm, or the, right, myofiber, inside of it is the myofilament, which is surrounded by the endomysium, and then in here, right, is our myofibril. Oh, it's going to talk about nerves. Right, we have our myofibrils, okay? There it is. Right, and then we have a sarcomere, and then we have actin and myosin. Okay, I'm telling you, the video is cold. Okay, so in our relaxed state, okay, we have that slight overlap. So the circles, the really crappy drawn circles. So the circles represent the myosin, and then the up and down lines, almost like the fence post, represent the Z lines, and then the other lines are the actin. So in our relaxed state, they overlap slightly, right? But they don't overlap all the way, okay? And so then during contraction, the thick and thin filaments slide past each other, causing myosin and actin to overlap more. Can we draw that? Okay. Let's watch, shall we? Okay. So this is in our relaxed state, right? This is actin, this is myosin. And this is where they're overlapping a little bit right here, but not a lot. So when it contracts, they're going to overlap a lot more because they're pulling the Z lines closer together and the sarcomere shortens. Okay, and so now it's going to shorten. So there's so much more overlap here than there was previously. Okay, so that's how we know the fiber is shortening and the muscle is shortening and we're contracting. Okay. All right. So the thick and thin filaments do not, none of the filaments change length at all. The only thing that's changing is that they overlap more. They're going to ask you on the test. They always ask this. They're like, do the filaments change in length? And you're like, no, they don't. They just overlap more. Okay, so don't get tricked. This one is like a big star. Don't let them trick you. Okay, so a cross bridge is when the myosin head is allowed to come up and make that connection with actin, right? We saw that earlier. This. Oh, it's kind of contracting. Right, this is that cross bridge when it comes up, when it pulls up and it makes that, and it touches the binding site. Okay. All right, so cross bridge attachments form and break lots of times. So each myosin head's coming up and breaking or grabbing different myosin, different actin binding sites and breaking and doing the same thing over and over and over again where it is as it shortens, right? Because it's going to get different pieces as it's coming together, okay? So they shorten a little closer so that the center of the sarcomere, and they move towards the center of the sarcomere in a ratcheting, right? Which is that little like piece like this, which is this piece. Right, it's also called the power stroke. It looks like ratcheting, that right there. Okay. Okay, so this is during contraction, right? Contraction causes shortening of the muscle fiber, right? The Z discs are pulled closer towards the middle, also known as the M line, right? The M line is just the middle. Okay, the I band, right, which is that part where it's only actin, shortens, right, because we're overlapping more, so we're going to have less of a place where there's only actin. Okay, the Z discs, right, also known as the ends of the sarcomere, get shorter or get closer together. Okay, the H zone, which is the piece in the middle where there's almost just myosin, right, where it's like the, right, so this is kind of like the H zone. Okay, it's like where there's not any overlap. Okay, that goes away, right, because it all overlaps, and so they end up almost touching. Okay, they'll end up almost touching, and so that piece goes away. Okay, and so the A bands, right, the part where there's actin and myosin move closer together and we form more A bands. It gets bigger. Okay, so let's see. So membrane, so all of this has to do with electrical impulses, because as our electrical impulses and our ion gradients change, it causes things to happen. So let's see. Let's watch. No. Okay. So the membrane, so the more permeable, depending on the size and solubility of charge, right, is our membrane potential, is how much potential does that membrane have to exchange ions, to create action potentials, and things like that. Voltage is the electrical potential to do energy based on separation. So the membrane potential is about the permeability of a membrane. So the more permeable depends on the size, the solubility, and the charge. So the amount of work or the amount of exchange of ions and things that can happen in that membrane to create an action potential, and so on and so forth. Okay. And then voltage is your electrical potential energy due to the separation of charge, right? So if there's more separation of charge, if there's a lot of charge up here and not a lot of charge up here, we're going to have a really high voltage because there's a really big difference in charge between these 2 points. Okay. So then an electrical potential energy, which is due to the separation of oppositely charged particles, right? So if I have a lot of negative up here and a lot of positive down here, right, there's going to be a lot of voltage or a lot of potential energy because there's a lot going on between them. Okay, there's a big difference. Okay, so charged particles, right, are the chlorine anion, the sodium cation, and the potassium cation, right? So K plus, Na plus, and Cl minus, right? These are the barriers that separate them in the plasma membrane. So these are the things that will be transferred between on the membrane potential. These are the things that are going to be transferred between on that membrane potential through the membrane, right, or the sarcoplasm. Okay, so change is also known as depolarization, which is conducted along the membrane, which is called an action potential. So let's see, where are we? Okay, we'll do this and then I'll show you how it goes. Okay, so there are 4 steps in skeletal muscle contraction, right? Nerve stimulation, so we have to have a stimulation to tell us to do something, right, which comes in that neuron, right? An action potential generated by the sarcolemma, right, which has a lot to do with the changing of ions, and we have an ion difference or a gradient or voltage, okay? And then an action potential is propagated along the sarcolemma, right? So in the beginning we propagate, or we have it, it's there, and then we propagate it along the whole muscle, so the whole muscle can contract, okay? And so then the intercellular calcium concentrations rise because, right, calcium binds to troponin, which will allow us to move that tropomyosin so we can make those little filaments move, okay? So let's kind of watch that. Okay, I'll put the closed captions on, but it's just better to watch it. Okay, so here's our muscle. So, right, so there's lots of, okay, right, so muscles have lots of nuclei, and they each have, it's called like a motor unit. It's like the muscle and the nerve and all of it. So this is the nerve or axon, right, that's gonna be attached to the muscle that we're looking at. Okay, so, right, so this, okay, is that nerve stimulation. Okay, so the nerve stimulation comes down the neuron, right, and it's signaling this muscle, right, and then it comes to this neuromuscular junction. I think we're going to talk about that later, right, which is just the neuromuscular, so neuron, muscular is here, and then the junction, so just the gap between them, okay? It comes down into here, okay? Synaptic bulb, okay, muscle fiber, cool. Interesting. Where did it go? Let's start from the beginning here, okay. We'll watch it. So, right, the nerve stimulation, and we're going to learn about this later. So this is acetylcholine, this little purple triangle, which will bind to these receptors on the inside of the muscle, right? So the light moving from here to here is when our action potential is generated, right? By the sarcolemma, which we said the sarcolemma is pretty much the plasma membrane for the muscle, right? So this is the action potential is being generated there, okay? And then it goes down, right? And this is it's propagating across the sarcolemma, okay? The initial piece was we have one and now we need to propagate it, right? So it goes down the T-tubule, okay? And then goes through the sarcoplasmic reticulum, which releases calcium and allows us to make these cross bridges and allows that muscle to contract and ratchet, okay? Okay, so the nerve stimulation and action potential occurs at the neuromuscular junction. NMJ is neuromuscular junction, okay? And then the action potential and the calcium levels rising is called excitation contraction coupling. So we're exciting the neuron, right? And we're allowing it to contract. So it's the coupling of us both exciting the muscle, and allowing it to contract at the same time, okay? Well, what did I do? Okay. So then we have nerve stimulation, okay? So the skeletal muscles are stimulated by somatic motor neuron. So this is a somatic motor neuron, okay? Motor for movement, somatic for body, and that's a neuron, okay? And so the axon is traveling from the CNS to the skeletal muscle. So the CNS is the central nervous system. So, okay, so, okay, so up here, right? It's traveling from your central nervous system. You can pretend your brain is up here, and then it's coming all the way down to the muscle, okay? So the axon travels from the brain, right, the central nervous system, to the skeletal muscle, and there are lots and lots of branches, right? Because each muscle or each fiber in this whole thing has to have a neuron that will tell it to do this, right? Because this is a muscle and then a fascicle, and then the little pieces that all go into it all need neurons to tell them what to do and when to stimulate, okay? So like, I don't know about y'all, but this blows my mind whenever I think about, like, every time I blink, or I'm sitting here doing this, you know, or every time I'm like moving my hands around, or anything I do, all of this happens simultaneously all the time, which I just think is crazy. It happens so, so fast. Okay. So the neuromuscular junction, right, is where the axon branches and ends at a muscle fiber. Okay. Which is just a fancy way of saying this, right? Axon branches and it joins to a muscle fiber, okay? That's the neuromuscular junction right here, okay? It's also called a motor end plate, okay? So how many neuromuscular junctions are per motor neuron? My boyfriend wanted to interrupt me during my thing. Of course, obviously, you know, part of being in quarantine. Okay. So how many neuromuscular junctions are per motor neuron? So there's one neuromuscular junction per motor neuron. So this is the neuromuscular junction. So that's saying that there's just one place where they attach, right? Each axon has one place where it attaches to the muscle. Okay. And so the axon terminal is just the end of the axon. So it's just, so, right. So this is like, right, CNS, and then we go down. So this is just the axon terminal. It's just where it ends, right? Okay, so the synaptic cleft is a gel-filled space between the axon terminal and the muscle fiber. So it's just right, it's in that neuromuscular, it's just in right here, okay, right in there. They just don't show you that, right? But the gel allows for conduction, okay? So that we're conducting things through that neuromuscular junction down into the muscle, okay? We can do it. I'm tired. I'm back. Super, super side note. I literally had to take a break during this session because I'm tired, and I don't even feel like I've been doing that much, but like I just feel mentally exhausted. So this is a reminder to take some time for yourself and do something that makes you happy, and do something that doesn't have to do with your schoolwork or your degree, or maybe that's by yourself or with some people that you love. Just do something that makes you happy, because that's really what matters in the end. You know what I'm saying? So let's get back to this muscle contraction, also known as how you hug people you love. Okay. Okay, so we were talking about there's one neuromuscular junction per neuron. So everywhere a neuron sits, there's only one neuromuscular junction, okay? So the axon terminal is the end of the axon. Oh, we already talked about it. So acetylcholine, right? We talked about that. It's that purple little triangle that comes out of the axon, okay? It stores in vesicles. I'll show you in a second. It's stored in vesicles in the axon, and it goes down into that neuromuscular junction, okay? It's stored in synaptic vesicles. Right? So it's a neurotransmitter, right, that tells the muscle to start inciting an action potential, okay? And the junctional folds are infoldings of the sarcolemma where they have acetylcholine receptors. So, right, neuromuscular junction, neuron. Is it going to show me any of it? Really not. Okay, cool. See, these are I was missing the last word last time. What is it? Synaptic vesicles, okay, and the little purple triangles are acetylcholine, and then these little orange triangles are the acetylcholine receptors. So, right, it's stimulated, synaptic vesicles come out, acetylcholine binds into these little acetylcholine receptors, right, and then they incite the action potential in the sarcolemma, okay? So what happens at the neuromuscular junction, okay? The nerve impulse arrives at the axon terminal, right, causing the ACH is acetylcholine, always. Acetylcholine to be received into the synaptic cleft, right? And then acetylcholine diffuses across the cleft and binds with the receptors on the sarcolemma, right? These little orange bits, okay? And then the acetylcholine binds to the and leads to an electrical event which will then generate an action potential through the muscle fiber. AP is action potential, okay? Which is that light that we keep seeing, the flash. Okay, so now we're going to talk about some weird stuff. So, homeostatic imbalances, when things don't work like we want them to work. So drugs and toxins can interfere with the events at the neuromuscular junction, okay? Myasthenia gravis, okay, is an autoimmune disease that's characterized by upper eyelid drooping, difficulty swallowing and talking, and generalized muscle weakness. So we're just having a lot of weakness issues, okay? This is because of a shortage of acetylcholine, and their receptors are attacked by their own antibodies, right? That's why it's an autoimmune disease, because their own antibodies are attacking their receptors, and so we don't have as much acetylcholine, so we're not having as good of a time doing things, simple things like eyelids, right? Like swallowing and just like general muscle weakness, okay? All right, so the action potentials across the sarcolemma. So the resting sarcolemma is polarized, right? Meaning the voltage is present along the membrane, right? Which we talked about. So there's lots of voltage and it's polarized because there's a lot of one thing up here. Maybe it's positive and there's a lot of negative down here. So it's polarized. There's different, like it's almost at poles right there at opposites. And so there's lots of voltage because there's a huge difference between these two, okay? And so inside of the cell is negative, okay? So then outside the cell is positive. So the action potential is caused by electrical charges, right? So a lot of negative inside the cell and positive outside of the cell. So we have 3 steps in this. We have the end plate potential, depolarization, and repolarization, right? So end plate, right? We talked about how the end plate is also the neuromuscular junction. So that's talking about the potential at that neuromuscular junction for things to happen. And then we have depolarization, right? Because we said the resting sarcolemma is polarized. So then we're going to depolarize it, right, to do that contraction, and then we're going to repolarize it so that we could do it again, okay? All right, so now we're just going to talk about everything I just said, and then we'll be done until Tuesday. Okay. Edit this out. Okay, so our first part is the end plate potential. So the acetylcholine is released from the motor neuron and binds to the acetylcholine receptors on the sarcolemma. This, okay, which causes chemically gated ion channels, also known as ligands, on the sarcolemma to open. Okay, so chemically gated ion channels. So it's a chemical gated ion channel. So it sounds like a big mouthful and you're not sure what we're talking about, but it is what it sounds like. Okay, so, right, which just allows us to open those pores on the sarcolemma, right? So then sodium diffuses into the muscle fiber, okay? And then potassium diffuses outwards, okay? And so then because sodium diffuses and it's less negative, right, it ends up making it more positive, okay, which results in a end plate polarization. A local, right, meaning it's in our space, like local, like Stephenville, not, you know, bigger picture like Fort Worth, okay, which creates an end plate potential. I think I have, whoops, where's my little, also, if we're going to talk about how there's, right, so I know this is kind of off topic, but I forgot about this, right? So this is like your spinal cord, right? If you took a cross-sectional area. So this is showing you how there's a connection to every muscle fiber, right? Or a large motor unit, right? It innervates a lot of muscle fibers, right? Which makes it pretty strong. All And then this is a contraction in full, okay? Right, so we're sending those waves, right? It goes down the T-tubule and through the sarcoplasmic reticulum, which is that darker blue, and it releases that calcium, which allows us to move the troponin and then move the tropomyosin and allow this myosin head, we're going to talk about this piece later, to, is it not going to show us contracting? Then, yeah, we're contracting, it's really fast, right? So it looks really slow in that other video, but it's contracting really fast, right? By shortening the sarcomere, Okay. What was it? Maybe I don't, that's fine. What you need to know about this is that we have chemically gated ion channels, okay? And then you need to know this bottom pit about the part about the sodium and the more positive, which allows us to polarize it. So read this a couple times, okay? And just really understand what we're talking about and how it's more positive and more negative. And you'll see that a lot in this video right here, okay? All right, so then we have depolarization, okay? Right, where you have a generation and a propagation of an AP or an action potential, right, which is like, we get to do something, yay, okay. Right, if the end plate potential causes enough change, so if there's enough stuff going on, then we'll reach the voltage gated sodium channels like threshold, which will open, okay? So if we reach the threshold, we end up opening that channel. And then, right, I think it's working fine. Okay, so then we reach that threshold, and so then the action potential can't be stopped. We've already hit maximum velocity and we're going, okay? So this will end up leading to muscle fiber contraction whether we want it or not, okay? So then the action potential will spread across the sarcolemma from one voltage gated ion channel to the next, causing a huge depolarization, okay? So this local end plate depolarization was like if we depolarized Erath County. And then this is like if we did the whole state, right? Because it's a pretty big jump. All right, so then we have repolarization, okay? Where after we've contracted our muscle, we restore that resting condition. And so the sodium voltage gated channels close, right? So the sodium stops moving and the potassium channels open and it resets and it puts the potassium back where it goes, sorry. Okay, so there's a efflux out of the cell of potassium, right, which brings the initial resting membrane potential to negative 70 millivolts. This is an important number, okay? Negative 70 millivolts is the resting potential of most muscle cells, okay? So this is a very specific number you need to know. And so then we're gonna learn about something called the refractory period, which is the specific time until we can repolarize fully again. So it's like when you it's like when you do a bunch of homework and then you realize you have more homework to do and you're like, all right, I need this much time of a break before I can do anything else, right? So that's our refractory period, is how much break time we need until we can actually be functional and do something else. Okay, let me make sure I showed you all the videos I wanted to show you. It looks like it. Okay, so I'm gonna edit this right now, and then I'll put it up. I'll probably put it up tonight, and it's gonna be like, do you really want to send that video? It's late, and I'll be like, yes, I do. Sorry, I'm really tired. But let me know if you need anything or if this material makes sense. Really, really, really watch those videos. I'll link the videos like up here or something. And if they're just in the playlist, I just recommend watching the playlist. It's really good, and it really helps you. Try the study sesh. Do something that makes you happy. You know, be kind to yourself. Be kind to those around you. And I will see you all later. I want to do the thing. Okay. A lot of toys in my desk. My boyfriend is pretty much annoying. Look, Anyway, that's not the point. Oops. All right, not the point. Also have